I've got bad news. You know that deadline you're worrying about? That thing that keeps you awake at night? The anxiety of just ordering food without making a mistake? It's killing you. It's slowly eating you up on a molecular level. It wasn't supposed to be like this. We were supposed to be huddled around a fire, telling stories about why the sun disappears every night. But now we got this! So what is this? The stress response is actually a pretty powerful bodily mechanism we've inherited from our ancient ancestors. It's designed to get us out of sticky, short-term situations. However, nowadays, our lizard brains don't know the difference between car horns and leopard roars, so our stress response gets triggered more often than necessary. But what is it? Stress begins in the brain, in your amygdala to be precise. When your eyes, ears, or imagination registers something potentially dangerous, the amygdala will recognize it for the potential threat that it is and signal the hypothalamus to do something. The hypothalamus is the part of your brain that controls all of the things you don't think about, like breathing, staying warm, how hungry you are, how horny you are, how fast your heart is beating, are you sleepy, all that stuff. It does this via the autonomic nervous system. This system consists of two subsystems, the sympathetic and the parasympathetic nervous systems. These serve complementary roles, with the sympathetic nervous system responsible for stimulating and preparing the body for physical activity, and the parasympathetic nervous system responsible for relaxing and maintaining equilibrium in our bodies. We return to our stressor. The hypothalamus receives the message that something's up, and it starts the stress response by stimulating our sympathetic nervous system. The main feature of our sympathetic nervous system is the chain of ganglia down and around our spine. These ganglia are connected to our spinal cord or directly with our brainstem, where they receive signals from our hypothalamus. Neurons between and connecting to these ganglia are called preganglionic. Extending out from these ganglia are much longer postganglionic neurons. Each of these wind their way through our body to connect to key organs. As our hypothalamus sends a stress response, our sympathetic nervous system activates and a signal is sent along each postganglionic neuron to their respective organ, where they release norepinephrine as a neurotransmitter. Norepinephrine binds with multiple alpha or beta receptors on the cells of these organs. These are GPCR proteins. If you are curious what those are, I discussed them in a previous video on the endocannabinoid system. But in simple terms, GPCRs change the inner workings of the cell when they are activated. For our hearts, that means faster and stronger pumping. For our blood vessels, that means constricting blood flow in unnecessary regions while increasing it in others, along with these other mechanisms. But this action is short-lived, enough to jump out of the way of a lunging predator or catch yourself from falling. This isn't always sufficient. We need more molecules to bind to more organs and tissues to keep them in a heightened state until we are comfortably safe. There are two mechanisms of supplying these molecules in abundance. There is the quick sympathetic adrenal medullary system and the slower hypothalamic pituitary adrenal system. The names sound more complicated than they really are. The SAM is the first to kick in, and this involves the hypothalamus sending a signal via the sympathetic nervous system to the adrenal glands, which causes them to release epinephrine or adrenaline from its medulla. The adrenal gland produces, well, adrenaline, or more accurately, catecholamines, epinephrine and norepinephrine. During the initial sympathetic activation of all the other organs, an unusually long preganglionic neuron extends down and into the adrenal glands. Did I say preganglionic? I did. Remember, postganglionic neurons release norepinephrine. If norepinephrine activated the adrenal gland, it would create a positive feedback loop where it creates the molecule that activates it. Therefore, the adrenal glands need the acetylcholine from a preganglionic neuron to activate. Once activated, they begin dumping epinephrine and norepinephrine into the bloodstream. Now the rest of our body can participate. But this isn't a great long-term solution. For starters, we are putting a lot of strain on our heart. It's pumping hard and our blood pressure is quite high. And we are using a lot of metabolic energy. Keeping this system running will burn us out. When times are less certain and our next meal is not guaranteed, needlessly burning energy is not a good trait to have. Therefore, evolution has learned the best thing to do is metabolize and remove catecholamines rather rapidly. So rapidly, in fact, that their half-life is only around 2.5 minutes. But stressors can last longer than a few minutes. How can we physiologically deal with those? Enter the slower hypothalamic pituitary adrenal system. About 10 seconds after the SAM is activated, the HPA starts. This begins with the hypothalamus secreting corticotropin-releasing hormone. This diffuses to the pituitary gland. Upon binding with the anterior pituitary, the gland releases corticotropin into the bloodstream. This then travels down to our old friends, the adrenal glands. Upon binding with receptors here in the adrenal cortex, this tissue begins to release cortisol into the bloodstream. This concludes the HPA circuit. The primary function of cortisol is to keep the body alert and energized, but cortisol reacts more intimately with our cells than catecholamines. Instead of binding to proteins on the cell surface like adrenaline, cortisol diffuses through the cell membrane where it will bind to a receptor floating around in the cytoplasm. After binding, this new complex will diffuse into the nucleus and eventually bind on specific sites on the DNA. These sites promote the creation of transcription factors, which then go off and promote the creation of different proteins depending on which cell cortisol enters. In our liver, cortisol causes an increase in gluconeogenic enzymes, or proteins that make glucose from other molecules. Which is a good thing, our bodies use glucose for energy. So if we need to escape a situation, we could use the energy. However, we need the base materials necessary to create this new glucose. The three primary precursors used to create glucose are lactate, glycerol, and amino acids. Lactate is a byproduct of regular metabolism. It's the molecule that makes our muscles burn after a good workout. Well, minus that acidic hydrogen. Glycerol is the backbone of all of the fats in our body. Our fats are stored as triglycerides, which are 3 long chains of fatty acids connected to a glycerol. And amino acids are, well, everything. Every protein, enzyme, molecular machine in our body is made of amino acids. The presence of cortisol in the liver initiates the conversion of these precursors into glucose to be used for energy. However, our liver is producing glucose for our entire body. It doesn't have the necessary reserves of these precursors to adequately fuel everything. Thankfully, cortisol has us covered. In our adipose or fat tissues, cortisol stimulates the creation of enzymes that split apart glycerides into glycerol and dampens the influence of insulin, so it's harder for adipose cells to take in glucose and lock it away as more fatty molecules. In our muscles, cortisol causes an increase in proteolytic enzymes, or molecular machines that break down proteins into amino acids. These glycerol and amino acid molecules, along with lactate from all over the body, will enter our blood and make their way to the liver. Now we can continue to create glucose to provide energy for our stressful situation. Cortisol not only increases the energy supply to the body, but also decreases energy use in non-vital systems, most notably by suppressing our immune system. Our immune response is incredibly energy demanding, but you already knew that. This is why you are bedridden during periods of extreme sickness. Someone recovering from a serious infection uses 50% more energy than they normally would. If you're unfortunate enough to have extensive burns on your whole body, your immune system will use every bit of energy it physiologically can extract from your GI tract. If conserving and producing energy is your goal, the immune system is not essential for the next 40 minutes as we deal with our current stressful problem. Suppressing the immune system has an additional benefit. It suppresses inflammation. Pro-inflammatory proteins can directly stimulate and sensitize peripheral nerves. This makes them more sensitive to pain. This is not useful when your life might end in the next few moments. In fact, it's kind of a distraction. Cortisol is decent at suppressing our sensitivity to pain while reducing inflammation. Sounds good, right? Well, inflammation serves a purpose. It helps our bodies fight off infection and bring in the resources necessary to clean things up. If wounds aren't inflamed, they aren't healing efficiently. Which is fine, we can lick our wounds when we are safe and cortisol levels die down. If cortisol levels die down. Before we go there, the last place cortisol influences that I want to discuss today is the brain. Specifically 3 parts: the amygdala, the hippocampus, and the prefrontal cortex. The amygdala is our emotional labeling system. When an event occurs, it will slap a label onto it, dictating whether it's a positive or negative thing. It's also where our fight or flight signal originally began. In the amygdala, cortisol causes an increase in brain-derived neurotrophic factor, or BDNF. This protein plays a crucial role in neuroplasticity and growth. Or, in simple words, BDNF helps our neurons grow and form more synapses. These extra synapses enhance the amygdala's functioning and makes it more vigilant and sensitive to potential threats. This can be an issue because one is at risk of creating a positive feedback loop. These effects are not short-lived. An acute high-stress event can cause an elevated BDNF level to persist for 10 days. The lasting impacts of a single high-stress event can be significant and may contribute to the development of stress-related disorders like PTSD. Our hippocampus plays a key role in learning and memory, as well as aiding in emotional processing. Its dominant function is to hold short-term memory until it can consolidate to long-term memory. For acute stress, norepinephrine suppresses hippocampal activity, effectively reducing background noise. This makes the hippocampus very sensitive to incoming signals from the amygdala saying, "Oh my god, this is bad, this is bad, this is bad," and helps ensure the emotional response to whatever just happened is strongly remembered. However, too much stress and you kind of only remember the emotional aspects. That is to say, you need a little bit of action to remember things. You can see that here. If there's nothing going on, there's nothing to remember. But when we are properly stimulated, we are remembering all of the details, and with too much adrenaline, we again start forgetting details and just remember the emotional response. So acute stress isn't too bad a thing in the hippocampus. But chronic stress has a different effect. Unlike the amygdala, stress and elevated cortisol here causes a decrease in BDNF. This causes a shrinking of dendrites and a loss of function and correlates to a decrease in memory and learning. Chronic stress also slowly reduces the hippocampal volume and additional cognitive functions. A reduced hippocampal volume is strongly correlated to clinical depression. However, of which causes which is still up to debate. Depression or not, chronic stress will slowly reduce hippocampal functioning and volume. The final place to discuss is the prefrontal cortex. Our prefrontal cortex is responsible for executive function. That is, making decisions and acting upon them. This is the part of your brain that tells you, we have food at home, and prevents you from stopping at a restaurant because we're feeling lazy. It helps us prioritize long-term goals over short-term impulses. Norepinephrine has a biphasic effect here, which means it has positive benefits in moderate doses, but at high doses it has negative effects. High doses of norepinephrine cause an opening of potassium channels, which moves neurons away from their firing threshold. Or, in simple words, it makes them harder to fire. This reduces the prefrontal cortex's functioning and as a result impairs higher order processing, allowing reflexes or lower order impulses to overpower them. Chronic stress and the presence of stress hormones, of course, negatively impact the prefrontal cortex. We see a reduction in BDNF as described earlier, as well as a degradation in the cytoskeleton supporting the neural structure. Near the ends of dendrites, or the receiving end of a neuron, are tiny protrusions called dendritic spines. These are the receptors of neurotransmitters and where synapses occur. Their internal structure is held up by actin filaments glued to the membrane by proteins called marks. Evidence suggests stress hormones stimulate protein kinase C, which then phosphorylates these marks. This causes them to detach from the filaments and membrane. Additionally, these filaments are held together by another protein. PKC also phosphorylates this protein, causing the actin filaments to detach and float apart. As a result, stress can cause dendritic spines to collapse and break synapses. An accumulation of these negative effects lead to a person with lower impulse control and difficulty concentrating as the functioning of the prefrontal cortex degrades with time. That concludes the brain, but we need to return to our previous discussion. Suppressing the immune system to conserve energy is a pretty good tactic. Reducing inflammation and suppressing immune responses might seem beneficial, but over time, our bodies are always trying to reach equilibrium. So this excess of cortisol causes lymphocytes or immune cells to reduce the amount of glucocorticoid receptors to compensate. This makes them less responsive to anti-inflammatory signaling, causing them to more easily release pro-inflammatory cytokines which activate the immune cells that cause inflammation. While inflammation is now running rampant, cortisol enters T cells and suppresses the signaling and production of interleukin-2. This protein helps our T cells multiply and differentiate into specialized subtypes and enhances natural killer cells' killing capabilities. This leaves our immune system on low alert, allowing viruses and other pathogens to accumulate in our bodies without a proper response and eradication. This is also why about 3% or half of us will often get sick when we are finally able to relax after a period of stress. The immune system wakes up, notices things are not right, and kicks into gear. I was going to end the script here, and there's like 20 other things to discuss. But then I found a whole nother issue that was fascinating to me. Remember earlier when I mentioned insulin gets nerfed in our adipose tissues? Turns out it's not just here. Stress can basically cause temporary diabetes, which may become not so temporary. One consequence of diabetes is steatosis, or the buildup of lipid molecules in organs. This happens in many places, but I'm going to focus on our muscles. Now that insulin resistance prevents our muscles from adequately fueling its activities with glucose, it begins taking up fatty molecules, or lipids, which can be broken down in the mitochondria for energy. However, excessive lipid metabolism produces reactive oxidative species, or bad molecules. These reactive species often target components of the mitochondria itself, causing dysfunction. Additionally, splitting lipids can produce ceramides and diacylglycerols, which interfere with insulin, reducing insulin sensitivity and causing the muscle cell to take up more fatty molecules for its energy needs. This negative response to the accumulation of fat or lipid molecules is called lipotoxicity, and the accumulation of fat in muscle cells is called myosteatosis. This accumulation is negatively correlated with muscle mass, strength, and mobility. Here we can see an example from a paper where these two women had the same lean muscle mass, but the one on the left here has almost double the intramuscular adipose tissue mass. The lady on the right had 45% more power in her legs, but this was not related to stress and was more focusing on myosteatosis in general. However, chronic stress can lead to myosteatosis. This in itself, along with other stress-related mechanisms, causes your lean muscle mass to decrease, which comes with a whole other batch of health complications. See what I mean about how crazy awful stress is? I didn't even get to these other organs or even mention all of the other critical systems in our bodies that just slowly get messed up the longer cortisol is around. But what I did mention was quite a lot. So, let's review. Stress has a dual nature when it comes to their effects on the body and brain. In the short term, stress enhances our survival by jolting our muscles and organs into action, sharpening and focusing our minds, and enhancing emotional memories. Our energy reserves increase, our immune system gets put on hold, and pain gets suppressed. However, the story changes with chronic stress. Over time, the same mechanisms that protect us begin to degrade. Although an anti-inflammatory, constant presence of cortisol causes T cells to compensate and become less sensitive to anti-inflammatory signals, paradoxically causing an increase in inflammation. Our T cells become less productive and our immune system becomes less vigilant to potential pathogens. An increase in insulin resistance causes steatosis or fat buildup in multiple organs, most notably our skeletal muscles. In the brain, the amygdala's heightened vigilance risks creating a cycle of hypersensitivity to stress, increasing anxiety and vulnerability to disorders like PTSD. The hippocampus, critical for memory and learning, suffers reduced volume and functionality, contributing to cognitive decline and potentially depression. Meanwhile, the prefrontal cortex, our hub for self-control and decision-making, loses structural integrity, impairing impulse control and focus. While stress can be a helpful ally in brief, acute doses, chronic stress is kind of a dick. It gradually erodes the foundations of our physical, emotional, and cognitive well-being. I know it's really easy for me to say this, but truly focus on the things in your life that make you carefree and embrace those. A lot of us keep pushing that moment further towards the horizon. If I can just get through this exam, give that presentation, get that promotion, or finish my thesis, then everything will calm down and we can relax. We forget all the times in the past we've made similar pledges to relax, but here we are. We're wired to focus on the stressors. It's unnatural, but for our own good, we have to focus on the things that relieve stress and bring peace to our lives.